are all essay questions, and I gave you all the essay questions, right? And so, I don't know, from the past, people who have wrote out the essays ahead of time and then studied that tend to do a lot better on that. If you don't, then you're probably going to walk into the exam and have no idea what to say. So, in chapter 1, we talked about some of the scientists during the golden age of microbiology. And one of the main themes in there was disproving spontaneous generation. What is spontaneous generation?
Life comes out of nowhere.
Something comes out of it, just appears.
Okay, so they noticed that organisms, bugs, or whatever, frogs, seem to just appear out of nowhere, and they just spontaneously generate, right? And so there were several scientists that worked to disprove this theory. Who was the very first scientist or scientific kind of guy to come up with the spontaneous generation?
To disprove it or come up with it?
To come up with the idea that everything is spontaneous. Aristotle. And so Aristotle was the first one to say that things spontaneously generated. But there were 4 scientists that we really talked about a lot. What were the 4 scientists? Pasteur, Redi, Needham, and Spelunzi, or
whatever.
yeah. Okay, which 3 actually proved that spontaneous generation should not occur?
Redi, Spelunzi, and Pasteur.
Okay, and what happened with Needham? He tried, but what happened?
They didn't have all the things you need to he cut off the oxygen and you need that for living organisms.
Yeah, he made some mistakes in the lab. He didn't seal his tubes well enough. Didn't boil the broth long enough, and there was cracks in there, so it got contaminated. So kind of really didn't help to prove it. So who took over his research basically and actually proved it?
Pasteur, was it?
Spelunzi. Okay, so what he did was he, number 1, boiled the broth for a little bit longer period of time to kill all the bacteria. He also kept his flask sealed and away from air, so that meant that that broth stayed sterile for a long period of time until you opened it up. Okay. What type of bacteria contaminated Needham's tubes?
Wait, what?
What type of bacteria contaminated his tubes, Needham? Because he didn't boil them long enough. Sometimes he used hay as a broth, which could be an egg.
Could be a spore.
A spore form. So spores are really hard to kill. You got to boil it for a very long period of time in order to kill them. He didn't boil it long enough, so those spores regenerated and contaminated his sample. So Spelunzi, he disproved spontaneous generation by being able to boil his long enough and keep his sterile. So then came along a guy named Redi. What did Redi do?
He experimented with decayed meat.
Okay, so they believe that flies and maggots spontaneously arose from rotting meat. So he did an experiment where he had a jar that had a piece of meat in there uncovered. He had another jar that had a piece of meat in it and it was covered with gauze. And then he had another jar that had meat in it, but he completely sealed it off. The one that was completely sealed off, obviously nothing grew on it, no spoilers really happened because that was sealed off from the air. The one that was covered with gauze, what happened with that one? On top of the gauze, but not inside it. So they didn't spontaneously come from the meat. The flies were laying their eggs on top of the gauze, and the maggots grew from there. Meat still spoiled. Why did the meat still spoil?
Cause it was out to there.
Air got to it, air has bacteria in it, right? And so the one that was uncovered obviously spoiled, and there was flies and maggots that were on that meat. So he proved that the flies and the maggots did not spontaneously come from the meat, but the meat still spoiled. So now we're still asking the question, well, why did the meat spoil? Even though the flies and maggots didn't get under. Because bacteria got it. We know that, but they didn't know that back then. So then you have Louis Pasteur. What kind of experiment did he do?
He experimented with the S-shaped vials to where the dust particles trapped.
Okay, so he had a flask and it had a swan neck on there, meaning that when he boiled his infusion, the air was pushed out of there, but then when he let it cool, the air was allowed to come back in. But we know the air contains bacteria, so in that swan neck, that hook of it, there was dust particles which also contained the bacteria. The bacteria was not allowed to get into the broth and cause contamination. So how long were these swan neck flasks able to keep their contents sterile?
18 months.
Yeah, for a long period of time, almost indefinite. Until you tip the swan neck flask over and the broth was able to mingle with the dust, and then when you tip it back over, the bacteria is now inside there, and after hours or a couple of days, it became cloudy because the bacteria was growing. So he proved that bacteria was present in the air, and that bacteria is subject to gravity just like we are, and when it goes inside that tube, it's not going to be able to come back up because of gravity, right? It's going to be trapped inside that loop. Okay, and so when you write your essay for that, I want you to be able to explain that in detail, because I do think I said, to name 3 of these scientists and describe their experiments. So you want to be able to describe their experiments and how it disproves spontaneous generation. Okay? Second question. You want to be able to explain Pasteur's experiment involving fermentation and how he used the scientific method to prove his hypothesis. Now in the past, I've asked this question and people give me basically 2 parts of what he did, but they don't explain the whole thing. The whole thing was, why did he do this experiment in the first place? Okay, so the vintners or the wine makers were upset that their wine was spoiled and they had a bitter taste to it. And so he did experiments to figure this out. And using the scientific method, first you got to understand, what is the scientific method?
Form a hypothesis.
Okay, you form a hypothesis after what? After an observation. You do an observation, that comes up with your hypothesis, so you have your question. Now that you have a question, what do you got to do? Test it. So you do experiments. So he did some experiments, and the first thing he did was trying to prove whether or not wine can ferment just from air. And so he boiled the broth to make it sterile. He left it exposed to air, but nothing happened, right? There's no fermentation. So then he boiled it again, because when you have your experiment and your experiment result are going to reject your hypothesis, then what do you have to do? You modify it. So he modified it, and then you retest it again. So he retested it again, and this time he kept it sealed. Nothing happened. Rejected that one, modified it. This time he added bacteria to it. What happened when the bacteria was added to it? Did it ferment? Is the juice fermented?
No. It was
Yes.
acid. Yeah?
Yes,
Oh.
because bacteria do participate in fermentation. It's just that the product from their fermentation is what?
Acid.
Acid. And so when you get this acidic taste to your wine, that's because the bacteria got in there and fermented and caused that acidic taste. So that hypothesis was rejected. Next, he put in yeast and nothing else but yeast and kept it sealed off in this walnut flask. What happened?
There was fermentation.
There was fermentation and it produced alcohol. Yeast during fermentation produces alcohol. Bacteria in fermentation is going to produce acid. So that's how he used the scientific method to prove that. Yes.
Were those left to air or were they sealed?
He had them in the swan neck flask.
Okay.
That was his thing, that swan neck flask. Okay? So you should be able to, explain how he used the scientific method to describe his experiments and how it works. Next one is describe Koch's postulate and how it proved the etiology of disease. Etiology is what?
The etiology of the process, the
Or
changing
cause?
of the disease.
Cause of the disease? Yeah. Okay, so what did Koch do? Hmm? What kind of animal?
Taught us the stages of infecting a healthy host.
Okay, so he had
With the diseased host.
the diseased host. The diseased hosts in his lab were mice. And so he had mice that had a pneumococcal disease. And so he took a sample from the dead mouse, and what did he do next?
He grew it.
He grew it in the lab. And so you should be able to grow it on, his postulates are that if you see a disease state, it should be the same disease state in all sick animals, right? And so they all have the same signs, symptoms, they all die. You take a sample from it, you should be able to grow it in the lab on a artificial auger, right? And so what do you have to do next after that?
To a healthy host.
No, you have to be able to isolate it.
Isolate
Just-
and grow outside of it.
Yes, isolate it and grow it outside the host. So that's what we do inside the lab. When we do our streak plate, we are, number one, growing it on the plate. Number two, the process of doing the streaking is to get an isolated colony. So I can take that colony, put it under the microscope, and I can see what that bacteria is. Each colony should only have one type of bacteria, right? And so if I can isolate that, then I know that every case is that same bacteria, right? And so you grow it, isolate it outside of the host. What did you do next?
And you infect a healthy host.
Yes, so now he had these healthy mice and he injected that same agent into the healthy mice. And all those healthy mice start to show the same signs, symptoms, and they all die. So does that prove that it's the same disease yet?
No.
No, because now I have to take a sample from that healthy host that I injected and take a sample from it and be able to isolate and grow the same bacterium that killed the mice. So I know that it's the same bacteria that caused the same disease in my first host and my second host, yes.
So he does it all over again? He has the mouse and then he takes the sample, then he grows it, whatever. Then he gives it to the other mice, then the mice die, then he takes the sample again and he does the whole thing all over
Yes,
again?
because you gotta prove that it's the same one. I can't just assume that it's the same one because what do we know about pneumococcal disease? What kind of signs and symptoms does it give to humans?